So, hello everyone. Thank you for attending the second part of the conference. So right now we're going to hear the invited speaker, Pankaj Bharmoria, and my name is Edwin Asradunas. I'm going to invite to present a little bit on his biography. So the Pankaj Bharmoria, he's a multidisciplinary expert. He is a biophysical chemist, as I might say. He finished his bachelor's in biology and chemistry. He has his Ph.D. in chemical sciences, and also he completed multiple postdoc positions all over the world, starting from Japan, Sweden, Portugal, and right now he has a position in the Institute of Material Science in Barcelona. So he's focused on organic molecules that can photoswitch, also in organic molecules that are capable to produce triplet-triplet annihilation. And specifically on this topic, we're going to listen to his presentation, and the floor is yours. Welcome, Pankaj Bharmoria. Round of applause for him.
So, thank you very much for the nice introduction. So my name is Pankaj Bharmoria. Maybe, I know how to start the presentation. Okay. Yeah. So my name is Pankaj Bharmoria. So I'm a researcher at Institute of Material Science, Barcelona, also known as ICMAB. And today I'll present my research on triplet-triplet annihilation mediated photon upconversion in confined nanodomains of biopolymer surfactant chromophore co-assembly. In the morning, you heard the lecture from Maria Abraham about the Tduc, and she theorized on the fundamental aspect of the Tduc. So I will show the work that I have developed over the years, especially the material perspective of Tduc. So basically, I come from northern part of India, this region known as Himachal Pradesh, okay? And my region is famous for two things. The first one is its natural beauty, and I invite you all for your next adventure trip to my state in India, actually, and you will be planning. This is the first part. And second part is, we developed the most of the hydroelectric power that is generated in India because we have a lot of rivers. So eventually, I am working on the development of, our state is working on the development of sustainable energy. And that is also the key motivation of my research, but in the other way around. So sustainable energy resources that I am developing is to harvest the sunlight. As we all know, the sun is the biggest external source of sustainable energy to the earth. And my research is focused on developing sustainable energy resources for enhanced solar light harvesting to increase the efficiency of the solar cells. So generally, these are the solar cells that we see around us, okay? But they can convert only 33% of the solar light that fall on them into electricity as per the Shockley-Queisel limit. And most of the other photons, they are lost either due to transmission, thermalization, or recombination. But the photons which are lost due to transmission, they can be utilized again if we use a suitable photon upconversion material, okay? So in that case, suppose if we use a very efficient photon upconverter just below the solar cells, we can upconvert the transmitted photons back to the solar cell. So in that way, we can increase the influx of the solar photon and increase the efficiency of the solar cell up to 44%. I mean, it is expected that photon upconverter material, they can increase the efficiency up to 44%, but it still hasn't been achieved. And I'm doing that in photoluminescent organic materials like this, using a process known as triplet-triplet annihilation-based molecular photon upconversion. So this system generally comprises of a triplet sensitizer, okay, which shows phosphorescence at room temperature, and the triplet annihilator, which have dark triplets at room temperature. So the idea is to convert or excite the molecule at low energy and get the high energy. That is why it is photon upconversion. So since it involves the triplet state, so the people who are aware with the photochemistry, they know the difference between the singlet and triplet state. You can see that the singlet state, the excited electron has a spin which is dissimilar to that of the ground state. That is why it is having an overall spin multiplicity of 1. On the other hand, in case of triplet state, when a molecule from singlet state goes to the triplet state, its spin remains the same. So that eventually results in the spin multiplicity of the 3. So overall idea is to convert the dark triplets of the organic molecule into the photoluminescent or photoemissive singlet state. So this is the general description of the TTUC. You can see it is comprised of generally it is comprised of an ensemble of chromophore such as triplet sensitizer, which I show porphyrin, and the triplet annihilator, which is generally organic molecule. And when we excite the sensitizer at low energy, it goes to the high energy triplet state by intersystem crossing, and while coming back to the ground state, it transfers its triplet energy to the annihilator molecule, whose triplets are generally present slightly below or slightly up to that of the sensitizer. And when two such sensitized annihilator molecules they meet, they undergo triplet annihilation that results in the formation of a high energy singlet state of the annihilator, and that shows the high energy emission via delayed anti-Stokes emission. Yeah, this is general picture of the TDUC. You can see over here the two low energy photons, they combine to form one high energy singlet photon. So this is a nonlinear optical process. However, to realize the practical application of the TDUC, we need to prepare the material which should be efficient. But to prepare the efficient materials, okay, for example, if you want to integrate them with a solar cell, they must function efficiently in the solid state. But that has many challenges. The first one is we use the photoluminescent organic molecules which have the aggregation tendency at high concentration. For example, if you want to use them for the biological application, then they must be stable in the solution state or aqueous solution. But generally they aggregate, okay? So that limits their application. And for example, if you want to use them in the solid state, so you dry the organic solution, they aggregate in the solid state as well, okay? And that results in the concentration crunching of the fluorescent. Another important aspect over here is the oxygen that we breathe that remains in the triplet state, in its ground state. So that is always available to quench the triplet of the sensitizer or the annihilator molecule. So we need to develop the material which could overcome all of these limitations. And that I'm doing in a soft material actually. So my research is generally focused on the development of TDUC material in the soft matrices. And over there I have developed some materials, for example, the photon upconversion hydrogels and the photon upconversion bioplastic as a semi-solid and the solid state photon upconverter to harvest the light ranging from green to the near infrared region. So actually I did my PhD from India in biophysical chemistry, and my PhD research was mainly focused on the surface active ionic liquid interaction with the protein. So basically I studied the interaction of surfactant ionic liquid with the proteins, and the important thing over here is that when we increase the concentration of the surfactant molecule, so with the increase in the concentration, they aggregate on the surface of the protein, okay? And when the protein molecule gets saturated, the surfactant, it forms micelles or vesicles in the solution. So when I went for my first postdoc to Kimizuka lab in Japan, I started working on photon upconversion at that time. So they thrown a challenge to me to prepare a aqueous stable photon upconversion material operating by TTUC, okay? Then, but for that to happen, we need to solubilize the dye in aqueous solution at high concentration. And over there I applied my PhD knowledge actually, because I knew that, okay, surfactant molecule, they aggregate on the protein surfactant at high concentration, and they form the aggregates. So we thought of the fact that, okay, whether these aggregates, they could solubilize this organic molecule at high concentration, because they act as a hydrophobic reservoir. And it turned out to be true. And we developed this nice work actually. So we use gelatin as a protein matrix, okay, and the diphenyl anthracene as an annihilator, platinum octaethylporphyrin as a sensitizer, and we use Titan X100 as a surfactant. So the surfactant role is very important over here because it's a non-ionic surfactant. On the other hand, if we use the ionic surfactant, they have the tendency to alter the structure of the protein, okay, and they also have the they have the chance of phase separating from the protein at high concentration, but that is not the case with the non-ionic surfactant. So non-ionic surfactant, they sits very nicely into the protein matrix and form the micelles. So into those micelles, you can solubilize this, what should I say, the organic molecule at high concentration. You can see over here from the confocal laser scanning microscopic imaging that these are the surfactant aggregates actually. They are containing these molecules, and they solubilize in the molecular form. You can see this hydrogel, it is very a transparent one. But the trick over here is that this molecule, which is in the high concentration, that should be ionic one. Otherwise, if we use a non-ionic molecule, it will aggregate even in the surfactant domain, and that system will not be that much of efficient. Well, we prepared this hydrogel because gelatin is a hydrogelator, so we use high concentration of gelatin. And that hydrogelation also acted as an oxygen barrier because, as I explained to you, that oxygen quenching is one of the limitations in Tduc. But this hydrogel, it functioned very nicely in the air. You can see we are exciting it with 532 nanometer laser, and it is showing the nice blue emission. And this is the steady state emission spectrum. You can see we are exciting at 532. And this is the phosphorescence of the sensitizer, so it is quenching. Okay. And in this system, we observed triplet lifetime of the annihilator to be 3.2 milliseconds, which is very, very long. Okay. And the upconversion efficiency, okay, I'm considering it 100% actually, according to it was found to be 13.6%, which was equivalent to the record value at that time. And this is a time resolved photoluminescence spectra. You can see that, okay, this hydrogel shows the triplet lifetime at two different concentrations. You see 3.3 milliseconds and 4.9 milliseconds. So the triplets of the annihilator molecule, they remain quite stable in this hydrogel. They are not prone to oxygen quenching. But why? Okay. And to understand the mechanism of that, we did thermoreversibility experiments of the hydrogel. You see over here, when we heated the hydrogel at high, up to 60 degrees Celsius, it formed the solution due to the disassembly of the gelatin fibers. Okay, because of that, the conversion emission decreased. It decreased because we prepare the hydrogel in the air. Okay, so when it is in the solution, oxygen has a, what should I say, oxygen can diffuse into the chrome 4 region, and then it can actually quench the triplet oxygen. But when we cooled it down again, oxygen is blocked. Okay, so it doesn't go into the chrome 4 region. So it retained its original upconversion emission. Yeah, this is, we also got these evidences that, okay, the gelatin, when it is in the gel state, it has this kind of a CD spectrum, which is typical of triple helix structure, okay? And when it is in the solution, it has a random coil structure. So when it is assembled, it forms a hydrogel. When it is disassembled, it forms a solution. And this strategy is not limited only up to gelatin. We also checked it with other biopolymers and other surfactant as well. For example, with agarose, which is also a biopolymer, and sodium alginate, and with other surfactant like pluronic F127 and twin-80. Okay, so this strategy is very much generalized. Then we go into the application part of this system, because if you want to explore them, obviously they should have some application. So the aqueous stable system, they obviously have the application in the biological system, because if we could develop the TTOC system that could function with a light in the therapeutic window that is ranging from 650 nanometer to 850 nanometer, then we could inoculate the material inside the body and then shine it from outside to induce a localized emission for a particular function. So we explored the application in optogenetic genome engineering. Okay, for that we prepared the a near infrared to visible photon upconversion hydrogel using tetratertiary perennial annihilator and this osmium complex as a sensitizer. Now the good thing about the pluronic is that it is a surfactant and it doesn't need a protein. So it can also form a hydrogel by itself at high concentration. For example, this is a hydrogel at 25% concentration of the pluronic in aqueous solution. And when we prepare the hydrogel and excited it with the 724 nanometer laser, it shows blue emission. So blue emission or blue light is very important over here. How? So now I'll explain a little about the optogenetic genome engineering actually. So we did genome engineering of the gene which correspond to the neuronal function of the hippocampal cell. So hippocampus is the part of our brain which has a major role in learning and the memory. Okay, but if somebody is having a neurodegeneration, then people can lose their memory. Okay, but that memory can be recovered, or learning process can be recovered if the neurons start functioning efficiently again. But how can that be done, and how our system is important over here? So what we can do is, we can genetically engineer the gene responsible for the expression of the hippocampal neuronal activity. So for that, what we did is, we use this photoactivable recombination plasmid. Actually, plasmid is actually extracellular DNA, okay, and which show response to the blue light actually. So it was transfected into this gene which is showing the activity of the neurons of the hippocampus, okay. And then, since it is responsive to the blue light, so we can excite the these neuron cells, okay, with the NR light, and we can expose this through the Tduc material that will be excited with the NR light, and it will show the blue emission. And this blue emission, due to this blue emission, this gene will be activated actually, and then it will show the neuronal activity. You can see over here, this is a diagram. We didn't do this experiment in vivo. This is just an in vitro demonstration actually. You can see over, this is the Tduc hydrogel. These are the neurons. And if you see, then, yeah, you can see the dendrite formation upon the excitation with the NR laser. Because of the NR excitation, it is the gel is showing the blue emission, and to which these neurons are responding. This is the biological part of the work. But if you want to integrate Tduc material with the solar cells, they should function efficiently in the solid state as well. Okay, yeah. So then I also started working on the solid state Tduc material. After my first postdoc in Kimizuka lab, then I moved to Casper's lab as a Marie Curie fellow, and there I started working on this photon upconversion bioplastics. Okay, because the Tduc material that we are developing, generally they are fabricated into the synthetic plastics. Synthetic plastic, okay, they can turn out to be plastic waste in future. So we thought of the idea that, okay, why not to prepare the bioplastics material? So, but how to prepare the efficient bioplastics? So we thought of the idea that, okay, if we could stabilize the viscous chromophore liquid inside the biopolymer, then they can isolate themselves inside the biopolymer like this, okay, and thick fiber network of the biopolymer that can protect the system from the oxygen quenching. In a way, for example, if this viscous surfactant liquid, what it will do is, since this surfactant will remain stable in liquid form inside the solid matrix, so that will avoid the aggregation of the chromophore, okay, and chromophore can diffuse very nicely, so they can function like a liquid state in the solid state, and they will be protected by the fiber network of the biopolymer that will protect from oxygen. Moreover, this surfactant liquid, it has a high boiling point, so it will not evaporate just like the normal organic solvent, and eventually it will lead into the formation of a sustainable solid state Tduc matrix. So working on this idea actually, what we did is, we took our hydrogel actually that I had prepared in Japan, and we thought of the fact that, okay, let's dry it in the air. How does it work? So when we kept it in air for 24 to 48 hours, all of the water that got evaporated, and that resulted in the formation of a very nice film. And when we shine this light film with a 532 nanometer laser, interestingly, it shows the Tduc emission in the air, okay? It is quite surprising, because generally, if you take any liquid Tduc solution and you put that into and evaporate the solution, the chromophore have the tendency to aggregate, but that didn't happen in this system. And then we check the upconversion properties like threshold excitation intensity, because when we are considering the integration with the solar cells, they should function efficiently near to the solar irradiance. So the threshold excitation intensity was 14 milliwatts centimeter inverse, and the upconversion efficiency was 15.6%. So interestingly, it was higher than that in the gel state, because in the gel it was 13.6% for the same system at the similar concentration. Another important aspect of this one was the durability of the system. So we checked this film after 2 years, okay, and it lost just half of its upconversion properties, okay. And then we elucidated the structure of the film, and we found out from the cross-section SCM imaging that, okay, the liquid surfactant droplets you can see over here, okay, they are stabilizing the chromophore. And from DSC thermograms, we also observed this is the peak of the, the melting of the Titan X100 with the surfactant that we use. From the dark field polarized microscopy, we found that, okay, this is a semi-crystalline film. So, that was also observed from the powder extra diffraction. So, which is very important from mechanical stability point of view. Yeah, so this film also showed the long triplet write time of around 2.5 millisecond. And then we checked the role of gelatin or the biopolymer in stabilizing to prove the stability against oxygen. So you see in the Titan X100 solution, this system shows upconversion emission, but that is not stable with the air. But for example, if you put gelatin into the system, form the film, so it is stable in the air. So it proves that gelatin is the main component over here that is protecting from the oxygen quenching. So this is the Tduc mechanism you can see over here in the biopolymer surfactant film. Yeah, since it's a liquid, so the molecule, they can diffuse nicely inside the solid state, okay, and nice upconversion emission. And then now, if you want to integrate them with the solar cell, we also need to consider the upconversion of the wideband photons. For example, we go to the NR region or the far red region. So considering that, actually we thought of expanding the upconversion window. But for that, we needed an annihilator, which should be ionic, and that should work well for the near infrared or red to a visible photon upconversion. So in that process, we could synthesize this sulfonated tips anthracene molecule, and then we used it with the different sensitizer molecule. For example, this PDDP-BP and osmium complex as sensitizer, which has triplet energy level that are quite near to that of tips anthracene actually. So the triplet energy could be easily transferred. And by this process, we developed the red to blue Tduc bioplastics systems, and you can see that it is showing very nice upconversion emission from red to blue. Okay, this is the steady state photoluminescence spectra showing the upconversion emission and the quenching of the sensitizer's phosphorescence. Eye threshold excitation intensity was found to be 95 milliwatt centimeter inverse, and it did show the record upconversion quantum build for the red to blue Tduc system, that is 8.5%. And then we also tested it to harvest 730 nanometer photons in the bioplastics. Yeah, so this is just a proof of concept demonstration experiment because we didn't explore it too much. Another important aspect over here was that now if we use these materials, what will happen to them at the end of life? Okay, will they go into the environment? And since we use the dyes over here which are toxic, okay, so they can pollute the environment actually. So to avoid that, we also developed the cloud point extraction process to extract the surfactant and the chromophore from the biopolymer, because biopolymer can be thrown into the system that will biodegrade, but we did not want it to throw away the surfactant and the chromophore. So for that, we developed the post-utility recycling process by cloud point extraction. This is a very simple process, because in that case, what you need to do is you need to put the sample at the cloud point of the surfactant. Cloud point is actually the temperature at which the surfactant gets separated from the solid state. So this Titan X100 reduced, that has a cloud point of 65 degrees Celsius. Okay. So when we put this sample for 30 minutes at 65 degrees Celsius, the surfactant and the chromophores, they got separated from the biopolymer very. Okay. Then you can do the decantation of the surfactant dyes, and eventually you can reuse them by adding fresh gelatin and prepare the film again, and it will show the photon upconversion again. By using this method, we could extract around more than 65% of both sensitizer and annihilator molecule, and this is the recycled film actually. So we use this molecule again to prepare a fresh film, and they showed the nice photon upconversion. So by doing that, I mean, it's also the part of circular bioeconomy, because we could develop that renewable energy capital, and we could also minimize the waste. So this work was highlighted actually at the front cover of the General Material Chemistry A. So finally, summarizing my research work. So we developed this chromophore surfactant protein co-assembly to prepare the hydrogel, which could function very nicely in the air. Eventually, we applied that for optogenetic genome engineering, okay? And finally, we developed the Tduc bioplastic for wideband photon harvesting, and now we are in a process to use them for the integration with the solar cell. So my impacts of the research, my research will have an impact on the current technology by developing the improved solar cells, and it will also give new direction for the plastic photonics industry to develop the sustainable photonics bioplastics. And moreover, my research will contribute towards the circular bioeconomy, because it falls under the main two principles of the circular bioeconomy, that is the development of the renewable energy capital and minimizing the waste by replacing non-renewable fossil-based products. And I think my research syncs pretty well with this principle. Yeah, because in the morning I saw somebody ask question related to the application of this Tduc material. And for that you can read our recent review article, because we have explained in detail the shortcomings of the current Tduc system and also the future direction which could be used for the integration of the Tduc material for the solar energy applications. Finally, I would like to acknowledge the people, first of all, the Vilnius University and the organizer of the conference to give me the opportunity to present my research work, my lab colleagues and the supervisor for their support, and I would like to acknowledge the different funding agencies like Science and Technology, Ministry of India, CSR India, JSPS Japan, Marie Curie Association, European Commission, and also the La-Caixa Foundation, Spain, who are funding my research right now. And eventually, Achula Bye. And now I'm open to answer any question related to this research talk.
Yeah, so thank you for the presentation, and now we're waiting for the questions. Okay, we have one.
Thanks for a very nice talk. So the first question goes back to the very first slide, where you told how much you can improve the solar cell efficiency.
Yes.
So this upconversion, in the morning session, we saw that the efficiency of upconversion can be theoretically up to 50%.
Yeah.
So was it regarding you have the best theoretical efficiency that you can achieve this 44%?
Yeah, with the upconverter, I think the best efficiency that could be, I mean the efficiency of the solar cell that can be enhanced up to 44% actually. Because the current theoretical limit is 33%, okay? But if you use the upconverter material only, not the down converter. If you use both upconverter and down converter, then you can enhance it further. But theoretically, it can be increased up to 44% if you use the upconverter only.
But upconversion efficiency regarding the best?
No, it's not the upconversion efficiency. I'm talking about the efficiency of the solar cell actually, using the upconverter. That can be increased up to 44%.
Okay. The next one,
yes,
quickly.
Yes,
please. So regarding, again, the solar cells, if you want to use low energy photons to convert to higher energy, and probably logically you would take silicon as best solar cell now technology. You would like to use it for silicon.
No. Actually, the Tduc-based material that we are developing at the moment, actually, they cannot be applied for the crystalline silicon solar cells because they have a band gap of around 1.1 electron volt. And most of the systems that are available in the system. Because in the molecular TDUC, you use the sensitizer and annihilator molecule. So the most of the sensitizer, they can absorb maximum up to 950 nanometer, okay? So that is above the silicon band gap. So obviously, they cannot be applied for the crystalline silicon solar cell. But whatever the proof of concept demonstration they have been done with a solar cell with the high band gap in the visible region. That is why people are generally focusing on the development of NI to visible photon upconversion. For example, the titanium oxide, okay, as a dye-sensitized solar cells, or the hydrogenated amorphous thin film silicon solar cell, and recently the perovskite solar cell as well. Okay.
Yeah, that's why was my following question. Like,
Yes.
if there are any materials that could be available to, or sensitizers, right, to absorb, I don't know, like 1,800 nanometers, and to convert it to the energy that is close to the silicon band gap.
Recently, actually, there was a paper by Professor Schmidt in Nature Chemistry, and they have used quantum dot as a sensitizer, which could absorb just below the silicon band gap. So maybe in future, actually, it is possible. But I think it is difficult to realize this with the molecular photon upconverter. But with the quantum dot as a sensitizer, it is quite possible, actually.
Okay, thanks.
Any more questions? Well, I have a few.
Yeah, sure.
So the gel structure that you use,
Yes.
the system, the gel system, it's quite interesting as it serves as an oxygen barrier.
Yeah.
How does it change over time? Does your gel somehow react with, let's say, maybe water vapor or oxygen and so on, and it changes maybe its absorption or something? How does it react over time?
Actually, since this gel is made up of protein, okay, and it also has a water actually. So if you'll put this gel, for example, if you put it in the air, okay, so eventually it will dry, okay. So because of the reason that, okay, there is a lot of hydrophobicity inside this gel because of the surfactant molecule, okay. So because of this hydrophobicity, water will always go away. So if you want to store this gel, you need to store them in the refrigerator at 4 degrees Celsius. And we did that up to 4 months. And after 4 months, actually, the efficiency was remaining up to 80%. Okay? Because it's made up of protein, so obviously you need to store them in the refrigerator, actually, at 4 degrees Celsius.
Okay, maybe there arise some questions from the audience.
And moreover, they are very easy to prepare, so you can prepare them in 1 hour, okay? So whenever you want to use them, you prepare them fresh. That's the best possible way.
Okay, then more questions from me.
Sure.
The recyclability is also very interesting, that you can recycle your films and use it again.
I don't know what happened, yeah.
What is the effect of recyclability? I mean, how does the efficiency change? Maybe your efficiency drops over time, then you decide to recycle your material, and maybe it comes back up, or how does the efficiency change when you recycle your film?
Yeah, you mean the efficiency of the TDUC film. Obviously, it will be lesser with the passage of the time, because if you continuously keep on hitting the film with the sunlight so with the passage of the time, these are organic molecules, so they are prone to photodegradation. But we are considering the recovery of the molecule which remains active. Okay, so that is why I said that, okay, we could recover up to 65% of the sensitizer or annihilator molecule in the active form. So accordingly, if you prepare the fresh film with this sample, I mean, you will get the same efficiency, although we didn't check in our paper actually, because I was supposed to leave Chalmers at that time. My contract got finished, so I couldn't do that experiment. So maybe, but we can do that actually. But that's a nice question.
And one maybe just from the top of my head, in each case for TTAP conversion, you use two materials, one sensitizer and one annihilator, right? Yeah. Is it possible to make sensitizer less upconversion in such a materials? Maybe just simplify everything and use one material?
Okay, I got your idea. I mean, in that case, for example, you need to integrate the, okay, I got your point now actually. So it is possible actually, and it was also demonstrated actually by chemical lab. What they did is they used anthracene molecule, they derivatized it with the bromine atom on two sides. So bromine is a heavy atom, okay, and when they excited it with the laser light actually, 730 and 630, it did show the upconversion emission. Okay, so it is possible sensitizedly photon upconversion actually, if you use a heavy atom. Although I haven't done it, and it is difficult to do, and you need very high excitation intensity for that.
So it would be a direct triplet absorber, right?
Yes. Direct S0 to T absorption in the annihilator molecule itself. But the issue with those systems is that they need very high excitation intensity, so it is better you use a sensitizer, because the excitation intensity is an important factor when you are considering integration with the solar cells.
Okay, maybe audience has questions. Okay, the questions arise.
Okay, so for the solar power, if I remember correctly, the number you presented, 1367 watts per meter squared, is the amount reaching atmosphere, and then the most is dissipated in the atmosphere. So my question is, whether and how precisely can you fine-tune the system to the atmospheric transmission windows?
Oh. I mean, I don't have any idea about that, because I took this number from literature actually, regarding this number. So your question is, how can I prepare a system to absorb maximum photons?
To use maximum from the sun power actually reaching the Earth's surface.
Okay, but actually that depends actually, because if you consider the AM 1.5 solar spectrum, so that varies from a different portion of the Earth actually. Okay, so accordingly you can devise your material. Okay, so it gets tracked actually with the movement of the sun. That is why, or otherwise there's another system that is like sustainable solar concentration. I don't know whether you have heard of it actually. So this kind of system, what they do is, so they change their track with a change in the sunlight, and according to that system, you can absorb the maximum solar light so that it can be converted into electricity in a maximum way. So your question is related to, absorbing the maximum solar flux.
Actually, the question is how precisely can you tune the wavelength absorbed so it's actually in the transmission window of the atmosphere. So when about 90 few percent reach the Earth's surface.
Okay, maybe I don't have an idea about that actually, because
Okay.
This is actually a problem related to the people in geology, maybe.
Okay, more questions? Okay.
Thank you for your presentation. It was really interesting for me. And the part was about genome editing.
Okay.
What kind of genes do those plasmids consist of? And how do they help to proliferate these cells?
Okay, maybe I can show you, because
it wasn't clear for me.
Yeah. Actually, for that, we collaborated with the expert who was doing the genome engineering, but maybe I can explain to you slightly. Yeah, for example, this Cre recombinase, actually it's a plasmid, okay, which is extracellular DNA, and which can be made photoactivable, okay. So if you transfect this into the gene which is responsible for the expressing the activity of the neuron of the hippocampal cell, in that case, what you will do is, when you will shine this gene, okay, with the TTOC hydrogel, you excite with the NR light, it shows the blue emission. And this, due to this blue emission, this gene will get activated and that will transmit the signal for the expression of the neuron to show the dendrite formation. This is how it is done.
Okay, so actually the cells that were used in this actually experiment was already engineered.
Yes.
Okay.
Yeah.
Thank you.
Okay. One question from me.
Sure.
I feel that it's time that I can ask one more question. So in various your research, you are using red or near infrared light to induce and obtain blue light.
Yes.
But sometimes you use DPA, sometimes you use perylene, sometimes you even use DPA modification. So various emitters. Why do you change those emitters? Why you need to maybe stick to one? Why to each time change an emitter?
Yeah, that depends on the sensitizer as well. For example, if you see in this particular system, okay, this tetra-tetrabutyl perylene, okay, so its triplet energy level, they are quite near to this one. So it also, the upconversion emission also depends on the triplet energy transfer from the sensitizer to the annihilator molecule. So it is very important you choose the right combination of the sensitizer and annihilator molecule to get the desired upconversion emission. This is the reason actually.
There's no preferred other quantity, maybe quantum yield or the emission wavelength? Or?
Yeah, that is also because, and specifically for a particular application, because if you use, for example, anthracene for this particular sensitizer system, okay, the anthracene may not work that well because the anti-shock shift will be on the longer side. Okay, because if you want to convert 730 nanometer into 480 and 730 into 430, obviously the quantum bit would be lesser. And there's another factor is when there will be triplet energy transfer from the sensitizer to the annihilator, the molecular orientation is also very, very important for the effective triplet energy transfer. Okay, because if you see for the dextran energy, for some molecule, they should be very near to each other, okay, within the 10 angstrom of the distance, and then they should have a perfect orientation for the effective triplet energy transfer. So for that case, what we do is we check different combination of the sensitizer and annihilator molecules, and those who works the best, actually we choose them for our work.
Okay, so there's an optimization process, right?
Yeah, obviously.
Each time that you need to think about, right?
Yes.
Okay. So, more questions. We have a lot of time. Okay.
I wanted to give all of the students opportunity first, but now I can't stop myself.
Yeah.
So I'm curious about all of your gel work.
Mm-hmm.
I know we see on the picture here that there is a little bit of scattering going on in that one, but
yes.
but actually it's pretty good, I would say. My experience, that looks like a very good film for upconversion. And this is, I guess, a speculative question, but we work with gels for both singlet fission and upconversion.
Mm-hmm.
And what we see is that in the singlet fission case, we have much less problems with scattering than we have for upconversion. And we've actually never done a gel that looks that good for our upconversion studies in terms of scattering. So can you just elaborate a little bit on how you study scattering and what you do to overcome that problem with the gel work?
Yes, actually, for example, if it's previous gel, so it's quite transparent, okay? Not this one, but the first one, emission. But for this particular gel, I mean, it was showing scattering, but we did observe the upconversion emission, which was significant enough to show the, to induce a response from the neuron, actually. Because in the Tduc system, high efficiency is required only for the solar cell integration, but that is not required for the biological application, okay? So efficiency is not a bar for the biological application, so that is why in this particular system, although this gel was cutting, but not that much of fire, okay? And we did show, we did observe actually a bit of scattering, but I think that wasn't enough to alter the upconversion emission, and you can clearly see that, okay, this emission is coming from the, what should I say, from the upconversion and not from the scattering actually. Yeah. Yeah, so this is also a learning lesson, because when you are considering the biological application, You do not need very high upconversion efficiency of the system. And also for the biological application, it is better that the upconversion efficiency remains on the lower side, because when you inoculate the system inside the cell, for example, then you need to use it for a function at a local level. So emission should be localized. Otherwise, if the efficiency would be on the higher side, then there would be a higher cellular autofluorescence. Because in the cell, there are many molecules which also absorb the light, for example, the visible light, and then they will start fluorescing. And they can also result in the auto-oxidation of these molecules. So that can induce the secondary pathways for the cell death. Okay, so if you want to use it for a therapeutic purpose, so the TTUC that induces a localized emission and is not very highly efficient, that is more useful actually.
Okay, do you have more questions?
Yeah,
Okay.
I'm happy to take more questions.
Okay, so we should conclude this part. Let's thank again the presenter.